So we start few final things. I could not present all those things, case studies. So there are few case studies about the specific enzymes which we have worked, and this is also some part of how the online tools can be helpful in generation of some new crucial data. So we told you about the xylanase. Xylanase are very versatile enzymes which are used for pulp and paper industry, and you can see that these enzymes are also useful for bleaching of the pulp. So what we did is that we tried to see all the xylanases from Thermomyces lanuginosus, and the study which we made was based on how this xylanase can be improved. So if any enzyme you are targeting, it can be improved by using specific tools. So what we did is that we did study completed the study of family 11 xylanases from Thermomyces lanuginosus. They are versatile enzymes, and we find that Y77W mutation can be better, and it can generate the xylanase, and it can make the xylanase in more precise way, means for the application part. So how we came with this idea is that during the wet lab study, we noticed that the xylanase which we are working is not much useful for the pulp and paper industry. So how to improve its capability, substrate binding capacity and all that. Along with that also we noticed that the xylanase which we are working is better suitable for the biofuel industry. So these aspects are always needed when you start your work in the preliminary phase, because after you work for 3 years or 4 years or 5 years and you came to know that our xylanase is useful for this industry, it means you have not seen the application part. So with the help of in silico tools, this case study will tells that this xylanase can be improved. So we did the molecular docking between this and different xylanase as substrates, and if you see, this is tyrosine 77 which is present in the active site. This is a very crucial binding module amino acid which gives you a better clue about that if you do the modeling and simulation or you generate the mutant based on this crucial binding area with this amino acid, you can get the improved capability. So if you see, this is the various mutants which we have generated online, you can see here, and these are the docking study with wild and mutant type of xylanases. So if you see here, in this we got this Y77F mutant, along with that Y77R and Y77W. So these are the crucial mutants which are generated through in silico tools, and they are telling that they are having a better capability and capacity for xylobiose and xylopyranose. These are two substrates used for the pulp and paper industry, also for the biofuel industry. So then we did the function prophecy, because this xylanase has lot of other functions also. So we did this function prophecy, and we know that this xylanase is not only useful for pulp and paper industry application, this is also can be used for other various application. And then we completed this study that how this xylanase can be improved, and this xylanase also other how it is compared with other xylanases from the mutant and wild type. And the whole study, computational strategy, is published. It is in the form of book, that is a Springer Briefs in Systems Biology. We are not able to study all the mutants, still we have the data for all the mutants, the docking, simulation and other strategies. So it is a small book, maybe probably 40 pages book. So it contains all the information about most of the family 11 xylanases from Thermomyces lanuginosus, and this gives a basis for the researchers who are targeting xylanase for other applications. Like these days, xylanase, use of xylan in also probiotics. you can see probiotics application, xylo oligosaccharides, you can see about that, okay, and also for the animal feed applications. So xylene is also used for animal feed applications. So there are a lot of angles which people are working, and the work is cited, and the best is that this is a thermophilic xylanase. So the versatility and applicability of thermophilic enzyme is better, because you can use them in the environment, direct in the environment as well. They will not be able to, they will not denature. So another thing is that which we did is that metagenomics, same enzymes. So I am presenting enzymes only. So we tried to do this sampling, and if you see, this is the cellulose binding module, and you see we worked on the beta glucosidase. This is also very good enzyme, especially when we talk about the biofuel applications, beta glucosidase. And if you see beta glucosidase, the activity and its profiling is limited, because sometimes we don't get the cold tolerant beta glucosidase. So our target was getting mesophilic as well as psychrophilic beta glucosidase. Psychrophilic means cold tolerant, okay. So what we did is that we did the samples from Kargil, and this study was conducted around 8 years back. Now we are compiling all that information into another module for that, but this study is very crucial, though we could not complete all the specific beta glucosidases, but we were able to get the clue that okay, cold tolerant beta glucosidases have immense capability for other industrial applications. So we collected the sample from the Kargil area, and also from the composting area, and also from the cow dung area. So you can see that KD, CM and CD, these are the codes given, and then we did the wet lab exercise, whatever we have done here, and then we tried to see the qualitative screening which we have done. This is published work, and here you see these are the isolates. Now our target was that what specific isolates we can take up further. So if you see, these are the different isolates which we got from various area, and then we identified the total number of microorganisms from Kargil was 7, and then compost and cow dung 11. So both one psychrophilic and one thermophilic. You can see that 50 degree and one 4 degree. So we had both enzymes, beta glucosidases. Then we did the carbon usage utilization using the biolog, and we also see that these species specific diversity was made on. Now if you see the isolates from Himalayan soil as well as compost and cow dung, you can see this BG8, along with that how the carbon source, different carbon source can be used for this. So when you design any strategy for utilization of the enzyme, what is the carbon, if you have a cheapest carbon source available, your enzyme production, enzyme capability will be cheaper, means you have the enzyme at a low cost. If the carbon source, nitrogen source is very high, okay, the amount is high or it is high quality, then the enzyme cost will be high. So our target is to use the low value substrates and try to get more enzyme out of it. It will give you the idea about that. So these are different substrates, you can see at 30 degree, 40 degree and 50 degree, and we are getting mesophilic, psychrophilic and thermophilic. So the sample collection has been done on different area to get different type of enzymes. Now we did this optimization. Here we have not used ANN tools because they were giving very good production, and then you see the profile of enzyme. I am not giving detail, but its application was then further studies. So if you see biological pretreatment, lignin removal, value added products. When we talk about lignin, so this beta glucosidase will be used, will be very useful through stain engineering. If you know that particular mutant, that can be used for specific lignin removal. Lignin has a lot of advantage, and one thing which I want to tell you is that whatever the UV creams you are getting, UV protection creams, you know that, which are very costly, they are using lignin material. okay, because lignin is not able to degrade fast. It is very tough to degrade, tough to break. Even you see these ceilings, okay, that also use lignin material. So lignin is a waste actually. So what people they do, we enzyme people, they remove lignin, okay, we do delignification. The pulp industry also remove lignin, because lignin will create impurities and the quality will not be good in the paper. So lignin is useful also. So we try to see that this lignin can be used for value added products, and also this uh, specific cold active cellulase production. Cellulase has lot of applications, so you can do that also. And this study we published, cold temperature active beta glucosidase from Pseudomonas lutea BG8, okay, and this was collaborative work with IERI, and Rameshwar was my PhD student, he is now in South Korea. And this is also the statistical optimization work which we have done. So what we have used is that paddy straw, that is the low value substrate. It is, I say it is a waste product, okay, which is thrown. So you can use this paddy straw, wheat bran, all these are the substrates which you can use. And paddy straw, you know, lot of problem of burning all those type of agricultural residues is a big problem in Delhi NCR. So that also gives the clue that, okay, you can use this paddy straw for cold active beta glucosidase, and this enzyme has lot of application in the saccharification, biofuel industry, as well as also for the lignin removal and lignin utilization later on. It is not only lignin is removed, you need to remove the lignin and then reuse the lignin. So these are the two I think I have covered this one, so I am not going detail here. Now as I discussed about this, the review articles, research articles and that, so this is one article, that is a forum article which we published about this PSBA in cyanobacteria. So this is, as I told you, the regulating the regulator. There is a concept regulating the regulator. How to do that? Okay, like if there is any regulation factor, how to regulate that regulation factor? That is a major challenge with the bioinformatics people. So we know that this gene is regulating the action of another gene, but how to regulate that gene is given, and what is the regulator there and what is the role of that regulator. So if you read this article, we talk about this isoform of D1, that is a PSBA, and it is a very crucial in PS2, photosystem 2 in cyanobacteria. This is one example, and it has also other cyanobacterium which are studied, but if you talk about this regulation, this also gives a lot of relevance for the other capability improvement in cyanobacteria. So in the same way, if you target any organism, so for this PSBA you can do this gene gene mapping as well as, and also you can try for the gene networking for this PSBA, not done yet now, okay, because the whole sequence, whole the transcriptome data you need, and this PSBA network you can identify along with that flux which is controlling the regulation. And what is the use of all this technique is that these specific cyanobacteria, they are controlling lot of other applications, like especially photosynthetic capability of the cyanobacteria, metabolite production in the cyanobacteria, and also the production of various type of other metabolites. This is also one review which I have shown you about bioinoculant capability, those who are working in bioinoculant and PGPR area, especially the plant growth promoting area. So you can use these tools given here, and here also we discussed flux which has not been covered in the tutorial because that was not part of this curriculum. So the flux part also you can think about. And then probiotics, those who are working, so these are the important aspects which you can see, and then system biology tools which can be used in this, and then probiotics of diverse origin and their therapeutic applications. those who are working in probiotic area, and see that how these tools can be used for various therapeutic applications. Foodomics is a new area, so we wrote this, we contributed for this chapter. Okay, so we talk about omics tools, foodomics. Okay, so lactic acid bacteria based omics, and this lab like study, day before study somebody was asking about screening and selection of culture independent. Okay, so the foodomics tools can be useful for the bacteria which cannot be cultured, unculturable. So this is also good. And then this is a new paper, a microbiome systems biology advancement for natural well-being. I told you about microbiome systems biology, microbiome engineering, and also precision medicine. All is covered in this tutorial review. You can read this, and this we collaborated with the two important scientists here. One is Donovan Antonia, and one was Joel Fantos. He is a doctor actually, he is in the gastroenterology department in Brazil. Okay, we also had one more collaborator, but he is willing to collaborate in this area further. And microbiome systems biology is a very emerging area and has lot of, so you read this review, those who are working in medicine and health related area, they should see, and there are lot of ideas I cannot discuss here. It's I think 15-20 page review, and you can read all these tools given there, and the tools are defined in this graphical abstract. You can see that precision medicine, drug optimization, personalized nutrition, healthcare, biomarker, and how these can be given. And this is the book which we have written. So this is all about the left out things which I could not complete. And I think anything left, कुछ और छूटा है? कुछ छूटा है? Anything left? Nothing? Anything was left? Huh? I just wanted that any of you should say. मुझे याद था, मैं सोच रहा था कि you remember then I will do. Paper writing, right? So I think Mohit, you missed that session, है ना? So you take the interval के बाद की story, interval से और आगे, ठीक है? So we will quickly finish this one. ये मैं करूंगा शेयर, इसमें नहीं करूंगा, अलग से करूंगा. हमारा वहीं पे जाते हैं जहां पे छोड़ा था हमने. एब्स्ट्रैक्ट, मेथड्स, सबमिशन, रिव्यूअर, एक्सेप्ट, रिस्पॉन्स, ऑथर रिस्पॉन्स. हाँ, दिस इज वेरी इंपॉर्टेंट. ओके, यहां थे हम शायद, है ना? ये भी हमने देख लिया था. रिस्पॉन्स, वेट फॉर प्रूफ, प्रूफ्स, एक्सेप्ट, रिवीजन, रीराइट सेक्शंस. ओके, यहीं से आपका शुरू किया है ना? यहीं से था, राइट? क्या और पहले से था? यहीं से था. ओके, सो you remember your recap the story. We talk about papers, how to write, what are the limitations, various reasons for not writing paper, and also how to improve the writing. Abstract, keywords, methods section, why it is important, introduction, what should be there, what should not be there, structure of the paper, submission, and result discussion, everything. Now if your paper is accepted with revision, means you got a revision. Okay, accepted with revision means after revision it will be accepted. So manuscript which are given revision, normally this is the first decision normally given for revision. So rewrite the section of the manuscript where it is confusing, because there is reviewer comments, you know that. So you can address all those comments and give manuscript to someone else to read along with reviewer comments. If you decide not to revise the manuscript, contact the editor and withdraw it. In case you have very big comments you cannot do, or there are comments which are really beyond the structure of your manuscript, or you have done HPLC, somebody is telling okay you get fresh HPLC sample and you don't have the time or you don't have the samples. So in that case you have to withdraw the paper telling the reason about it. If you hold it for 1 month, karenge and it doesn't happen, it doesn't look better, because next time when you submit the paper they will know this is the author who has not submitted the manuscript on time and also not informed us that we are not going to submit it. So please remember this. Response to this I did once in my life, this one never I did in my all 24-year career. Once in my life I did 2 years back with one of the paper where some additional experiment was required and my student was not available for the experiment. So she got some other family engagement and she was not available. So I wrote to editor that okay, we will withdraw this manuscript, we don't want to continue with it. Manuscript is still with me, okay, because additional experiment is required, because it was a preliminary information. So sometimes journals they ask more data. So you have to be very honest in that. If you don't have the data, you cannot submit it, okay. So it is better to inform the editor. Normally we leave it, okay, kya hota hai us, but you have to inform it. So maybe you get this, you should not get this type of thing, but in case you have some additional experiment to be done and the person who is supposed to do is not available, then you have no option other than taking it back the paper, that okay, I will do it whenever our student is ready. Now response to the letter, this is very important. Most of the time this is a tough task. I think you see that sometimes very tough questions, sometimes supportive questions, sometimes tricky questions, sometimes no the question is having no clue what to answer it, okay. Sometimes philosophical question also. So you have to see that you can do the additional experimentation or data if analysis is needed. For that the journal normally gives 3 to 6 months time if you need to do some additional experiment. Some journals they give more than 6 months also if the experiment is long. So you can take time, no problem about it. Make appropriate revisions in the manuscript, point by point response to the comments given by editor and reviewers, and when you make the response also put the line number where you have done the changes. That is very important. And then respond to the letter and the intent of the suggestion, explain any additional changes you have done and return the revision in the timely manner. Normally they give 21 days to revise the manuscript, but sometime it takes more time, because 21 days तो बहुत fast निकल जाते हैं. You can ask additional time. You can ask the additional time of 1 more week or 2 weeks or 3 weeks, but ask once. When you want to do revision and you want additional time, don't ask the time by which you cannot complete. So you have to ask at once. ऐसा नहीं कि you ask 21 more 2 weeks, then you realize I require 1 more week, then again you realize I require 1 more. Don't do that. You estimate and take 1 week extra. If you require 3 weeks, you ask 4 weeks. If you require 4 weeks, you ask 5 weeks. So this is a strategy which I am telling you. And you should always tell your mentor or your supervisor the exact date by which you can give, so that the same can be sent by the mentor and supervisor, because this work is done by mentor. So they communicate with the editor, nobody else can do that. So it is very important to estimate the correct date of revision submission, and always take 1 week advance so that you have some time in hand. I will say 10 days, 1 week से भी ज़्यादा, 3 दिन और, ठीक है. So 10 days आपके पास होने चाहिए, and you should not wait for the deadline. If the deadline is say 15th of June, so your work should be ready by 7th of June. so that you have additional 1 week time for your supervisor. Otherwise you send it and then you say sir submit nahi hua, kal to last date. So this is not a good practice, and many times I have seen the students they do like this, okay, and you recall and you will laugh at yourself, okay, that I also did like that, okay. So please don't do that, because now after this Gyan program, this is an accessory session which is very important for you. Then more on rejection, okay. If your paper is rejected, I told you don't take it personally, that paper is rejected and I am very unhappy, don't take it personally, okay. It is a science. Look at the good points and build on them. Editor can help to resolve the conflicting reviews. If there are one positive review, one negative review, but the editor took the decision based on negative review and paper rejected, so you can still talk to editor and they can write that okay, this was the comment, can I get the revision or something. Don't pretend to fix something if you haven't. Then if sending to a different journal is the previous rejection was relevant, be diplomatic. This is a very important thing. Don't do any resubmission without improving the previous rejected paper. These days with AI, if you submit a paper to Elsevier journal, another Elsevier journal will get the data that this paper was rejected from X journal or Y journal. So you don't be smart that okay, this journal will not know that the paper is rejected, and the comments are also forwarded, okay. So if again the editor will look whether they improved it or not. So improve the paper, don't submit it happens. Sometime you see one manuscript rejected in one journal, again rejected, again rejected, again rejected, you get repeated rejections. Why? This is the reason, because you don't improve it. So be diplomatic, don't take it personally. Why a manuscript get rejected? I told you in the beginning only, inappropriate journal. Research is only confirmatory, study is incremental only, study is not designed properly, conclusions are not supported by data, manuscript written poorly written or organized. These are the major reasons of manuscript rejection. So if you see these reasons, you have to improve it. Ethical considerations are also important, because integrity is very important, so you have to do that. Coming to the authorship, I told you don't make anybody author still, and they don't contribute effectually, so you have to have the idea about the authorship. So when it is decided who are the authors, who is an author, what are the responsibility of an author, how is decided what is the order of author, what are the career implication of the authorship position. These are important questions and the answers I will provide you. Okay, the requirement of authorship is contribute to conception, design, acquisition, data analysis, then involved in drafting and revising manuscript, approve the final version. These are the three responsibility of the author. Okay, an author must be willing to take public responsibility for the content of the manuscript. Being the author is not easy. If you are author, you have to take responsibility of the work, whatever is reported. So this is a very important and challenging thing. Whatever we are presenting here, we have to take the responsibility of that work, that okay, this work is done in our lab by this particular person. ऐसा नहीं कि जब कुछ आपको गड़बड़ हो तो आप कहें no, I don't know. Okay, so you have to take the responsibility. So whatever you are reporting to the world, you are responsible. So report it once it is verified. Okay, don't make it like a breaking news. Okay, you have to see that this is your responsibility. Then author responsibility, defend the intellectual content including results and discussion. Be willing to concede publicly if any error is there in future, you should be able to accept and correct. So these both are the things there. In case of fraud, publicly taking the responsibility is to be there. So normally it should not happen, but if it is there, the author also have the responsibility for this, that if you are reporting anything, there should not be any misleading content in the manuscript. Okay, then going further, authorship listing, decide very early. Okay, I am telling you, authorship sometimes creates lot of problem. Mohit might have experienced during PhD, because he has just 2 years back, 3 years back he has done, and few people are doing PhD. So this is a very pertinent issue, authorship. So you decide early who will be in your team. Okay, 1, 2, 3, 4. Don't make a team in the last, because in the last sometimes some people will contribute, sometimes will not contribute. But those who are in your team, they should be asked to contribute. In case some specific editing is required, you can take another person in the team as your editor or the proofreader or scientific help, okay, for some sections, connections, okay, or senior authors to help, okay. Principal investigator is the last author. Lead experimenter listed first. Dual first authorship is possible, okay. Sometime it happens that the lead experiment, if the experiment was done by me, I should be the first author, okay, 70% of the work, okay. Normally it is followed, and the mentor, supervisor, professor and principal investigator goes to the last, okay. So authorship problems, first author and PI don't agree who should be the author. These are the few problems I am just telling you. PI includes, deletes author without discussing with the trainee. What do you do if you left off as an author? What you will do? Like you contributed, but आपका नाम नहीं है paper में. So what you do? Then how do you handle co-workers asking to be included in the manuscript? Sometimes it is a big problem. Okay, you worked with 2 people, they helped you, and your supervisor, mentor, and you didn't made him her as a co-author. Then what do you do as a secondary author and you have concerns about the manuscript content? So these are few questions. So you have to follow the ethics and try to resolve all these issues, and this is important for the public trust, proper use of funding, protect research subject, health and rights, ensure accuracy, reliability, collaboration and scientific progress, give people the credit. I told you about the acknowledgement. This I already told you. Plagiarism I am not covering because it is already there, but don't plagiarize. I told you, don't plagiarize, write yourself, don't use too much AI tools, okay. Use AI tools in your research, especially when you design the experiment, you try to find out genes, use AI there, don't ask them to write for you, okay, write yourself, okay. There are 3 types of plagiarism which is there: mosaic, paraphrasing and self, okay. So all 3 are not good, they are bad, so please try to avoid that, okay. Self plagiarism is also there, okay. Sometime you don't do that, okay. Others will cite your work, why you self cite it, okay. You can cite 1 or 2 papers from your group which is very relevant, but don't cite too much from your group, okay. Cite other important study, okay. So self plagiarism is also not accepted, okay. Guidelines, scope guidelines is there. The common problem of authorship, one exercise: did you come across conflict among the group members resulting from authorship issues? Many times you have, okay. So you can remember your own thoughts and try to see that. This is one exercise, and if you get through these aspects, you can definitely understand that what is your contribution, how the authorship is decided. Types of paper, I have showed you all the papers: review article, editorial, book reviews, letter to the editor, book chapter, lay writing, okay. Except lay writing, all these are called as publications. Sometimes people they confuse that review article is not a publication. No, it is all called as publication, okay. So review article also very important, it will helpful. Research article, editorial, book reviews and others. Review article, it is not original. What is review article? I have already shown you. It is like a tutorial paper where you cite various type of research articles and get knowledge about that. Review article have a wider audience. The writing style is very general, and it is the writing will be extensive and it will be expansive also. Introductory paragraph, very important. Conclusion, important component of review article, and it can be painful because writing conclusion on a bigger way is very difficult. may influence the future readers. Open writing, editorial and book reviews. So normally the editorials and book reviews, they are given to the senior authors, okay, including the review article. So then editorial is perspective. So I am not going detail here. Book reviews written by the senior authors. Letters to the editors. This is also written by senior authors, not by that. That is also publication. You can see in Science, Nature, other journals, letters to the editor is also there. Okay. Book chapters. Book chapter mostly I think many times you write. So how to write a book chapter? Please note that. Write it well, submit it promptly, follow instruction. Can be like a review article. Plan the chapter carefully, organize and meet the deadlines. Normally chapter deadlines nobody follows. Many times it is book types. It.org resources writing center. It is given here. Okay. So this is a very important. I think any of you have visited here. Jyoti, you visited here? Huh? Okay. So this is a very good resource center, and it gives you an idea about how to write discussion, conclusion, great title, everything is given there. So these are few reviews which are highly scientific. I wanted to show you here, and these are collaborative reviews. This is another review, enhancing the production of biopigments, then gene editing, then this is a book. This I already shown you, so I am not going detail here. So I think so. Thank you. I think I finished. And so this is very important when you complete any course like Gyan academic curriculum, to understand that whether this study which you are completing or doing after taking the techniques idea from this course can be used. Okay. And secondly, how to make your research paper, that is very important, because data analysis, data presentation, these are two different things. How to present the data, how to consolidate the data, that is very important. Okay. So with this we close the session now, and if you have any questions we can discuss it later, and we are available tomorrow also. If you want to discuss anything, we will be able to help you with that. But normally the course concludes today with this session, and we have already taken your examination. So the grade sheet and certificate you have to wait here, and we will be soon giving you that in the break of maybe 10-15 minutes after the tea. Okay. So the tea session is there. After that we will try to give you the